and welcome to what I've decided to dub the Made Easy series. In this episode we'll cover open pollinated cultivars, F1 hybrids, Pureline cultivars, and integration made easy. Since open pollinated varieties and Pureline variety are often used interchangeably and to mean different things, I'll separate them into two different categories. In perennial crops, open pollinated cultivars are cultivars created when one knows the maternal parent but not the paternal parent. Let us take for instance Golden Delicious. This apple cultivar was raised from a seedling of an apple tree cultivar called Grimes Golden. Its pollen parent is unknown. A great many apples of the ancient world were of this type. In essence, basically finding a tree you liked and planting seeds from it. It was an old way of doing things back in that time period, and to a certain extent it still gives great results today. Now on to Pureline cultivars. When it comes to Pureline breeding, the basic gist is you first make a crossbreed between two self-pollinating species using artificial methods. Take wheat for instance. When you cross two specimens together, you get an F1 hybrid. This progeny is very vigorous and very prone to variation. You let that then crossbreed with. What happens after the crossbreeding process, you select one of the progeny that has all the traits you want and collect seeds from it. With each generation that passes, by repeating this process and only selecting the plant that's identical to the previous one, the amount of phenotypical and genotypical fixation that happens increases each generation. After selecting seeds from one individual plant, which is self-pollinated, 7 times in a row, all the traits you've looked for in a wheat variety have become genetically fixed, almost completely. After the 7th generation of self-feign and selection, 99.7% of all progeny produced from this plant will be genetically identical. This is what we call a Pureline cultivar. Most wheat, soybean, and cereal crops are amongst this type. This is just the simplest way of doing things. There's several other ways that can build upon this process for wheat breeding, or Pureline breeding in general. This diagram was made by the late Dr. Ali Navabi, one of the most important wheat breeders in Ontario, and a great teacher. But I digress. This image lays out the ways to get an F1 hybrid from which a new Pureline cultivar can be selected from, such as crossing the initial F1 cross with another parent, or crossbreeding two F1s together, or continually breeding back the F1 into one of the other parents to increase the amount of pedigree from that parent, before going over the process I had laid out before. Another method is through double haploid culture. This is where haploid cells, i.e. anthers, of the male parts of the plant are collected and tissue cultured. Afterwards, chromosome doubling chemicals applied to the cultured plant material to make it viable. These are called double haploid varieties, and as a result of this process, each seed they produce from self-pollination will be genetically identical to the parent plant, with no variation available. One can create a Pureline cultivar very quickly using this method, although you lose selective variability to draw from to make new cultivars. Now let's talk about F1 hybrids. F1 hybrids are where we try to capture the vigor of an open pollinated hybrid with the vigor of a heterozygous strain. This is done by crossbreeding two known Pureline cultivars together en masse and mass producing that seed. F1 hybrid seed is still genetically to each other, so every seed is genetically identical from these initial crosses. To that end, Pureline cultivars are maintained for crossbreeding purposes. This only works in crops that are easy to crossbreed and mass produce. The best example of F1 hybrids being used would be corn. Now on to integration. Like with backcrossing, integration involves the hybridization of two different species, or two distantly related strains of the same species together, then crossing back into the species you want. If you take the pedigree of Gold Rush, for instance, the original parents are the domesticated apple variety Rome Beauty and a wild crab apple called Malus floribunda. The next generation is the crossbreed between two sibling plants born from that union. The third generation is a hybrid between the result of that union and Golden Delicious. The fourth generation is a crossbreed between a domestic apple called Melrose and the results of the previous union. The fifth generation is a cross between the domesticated apple Illinois 2 and the progeny made from the previous generation. And Co-op 38, better known as Gold Rush, is the crossbreeding of the results of all those generations being crossbred with Golden Delicious. As you can see, through frequent back crosses back into the domesticated apple germplasm, most of the crab apple DNA has been bred out, leaving behind only small trace amounts, including the VF gene, which imbues resistance to various scab strains to the progeny. Well, that about covers everything. Thank you for watching.